Hello, welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding the effect of temperature on the rate of enzyme activity and its impact on plants, animals, and microorganisms. So what are the optimum conditions for enzymes? So enzymes are tertiary structures that are sensitive to changes and require optimal conditions to keep functioning. Changes in the environmental conditions can lead to the denaturation of a protein or enzyme. This means that the protein or enzyme has lost the shape of its active site and can no longer function. So we will be exploring enzymes in a lot more detail in topic 2, which will be uploaded soon. But this is all you need to know for topic 5. So changes in temperature. So an increase in temperature is going to lead to an increase in kinetic energy. So more frequent collisions take place, which result in more successful collisions. It's quite good to know these 4 points because they come up as 3 or 4 marker questions. So this is basically the bullet point that you need to know. So, however, a temperature increase or decrease that goes beyond the optimum can result in the bonds in the active site breaking and the enzyme loses its tertiary structure. The specific shape of the active site is altered and is no longer complementary to the substrate. So what is the temperature coefficient? This is a calculation that you will be expected to do. So the temperature coefficient is the degree to which the rate of reaction changes with temperature. So it can be calculated using the following formula. So Q10, that's the temperature coefficient, and R2 is the rate at the higher temperature, and R1 is the rate at the lower temperature. Both will be given to you in your exam question. So what does the temperature coefficient mean? So a Q10 value or temperature coefficient value of 2 indicates that the rate has doubled. A value of 3 would indicate that the rate has tripled, and so on. So now let's look at the changes in pH. So different proteins and enzymes work best at different pH values. So pH values that are away from the optimum pH of the specific protein can once again lead to denaturation. This happens because the ionic and hydrogen bonds in the tertiary structure break and the active site changes shape. So now let's look at substrate concentration. So initially an increase in substrate concentration can lead to an increase in the rate of the reaction. However, after a while, the enzyme active sites are saturated and the concentration of the substrates are no longer the limiting factor. So what that basically means is that all the enzymes are bonded to some substrate. So an increase in substrate is not going to make a difference because there are no more enzymes to bind to. So this point is called the saturation point, and when plotted on a graph, the rate of reaction after the saturation point levels off. So now let's look at enzyme concentration. So initially, just like substrate concentration, an increase in enzyme concentration increases the rate of reaction initially. However, after a certain point, the concentration of enzymes is no longer the limiting factor and there is no further increase in the rate of reaction. So same way as substrate concentration, after a certain point, there are no more substrates that you can bind to. So increasing the enzymes is not going to make a difference. So that's why, just like substrate concentration, if you plot it on a graph, it's going to level off after a certain point. So measuring the rate of reaction. So we can calculate the rate of reaction by taking samples from the chemical reaction at different points of time and measure the change in concentration of the substrate or the final product. So immobilizing enzymes. So when enzymes are immobilized, when they are used commercially, and this makes it easier to separate from the product, easier to reuse and reduce the cost of the production process. They also have higher resistance to changes in temperature and pH, hence minimizing modification of the active site. So I would say this is quite a good slide to be aware of because I haven't seen that many questions on this, but it did come up once and it's not very clear in your textbooks either. So I would say know this slide off well, understand it pretty well. Looking at enzyme activity and living organisms, so photosynthesis is essential for the converting of carbon dioxide into carbohydrates. This involves proteins in the electron transport chain and enzymes such as ribisco, which we have previously explored. So if you're not too sure, do go back on the topic 5 playlist, which should be uploaded by now. At high temperatures, plants carry out a reaction called photorespiration at a faster rate. However, this reaction also uses the enzyme ribisco, hence the higher temperature lowers the rate of photosynthesis, hence reducing crop yields. Some fish eggs also develop more slowly at higher temperatures. The sex of the young in the eggs of some species, like turtles and tortoises, is controlled by temperature. And migration patterns of animals might change to cope with the changes in temperature. So that is all from Professor Snap today. Hope you found this topic useful. If you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.